All life is made up of 4 organic macromolecules: carbohydrates, lipids, proteins, and nucleic acids. Each of these macromolecules is made up of different elements. All of them include carbon, hydrogen, and oxygen. BRCA is a gene, which is made of nucleic acids, that produces a protein. This protein screens for DNA damage and repair. In order to properly function, all proteins must be folded in a certain way. This folding is determined by the amino acids, which in turn are determined through transcription and translation of genes. Unit 2 describes the general structure of cells, including the major differences between prokaryotic and eukaryotic cells, one of which being that eukaryotic cells have membrane-bound organelles. In connection with this lab, scientists target proteomes lining the cell membrane. These proteomes are complex proteins produced by the cells, in this case, located in the cell membrane. Proteomes are targeted by specific personalized cancer treatments. This prevents BRCA problems by stopping cell division before tumors can occur. The picture illustrates how a drug might inhibit the genes allowing for cell division within cells that have a mutated BRCA gene.
Unit 3 is all about cellular energetics. Cellular energetics is the study of production, distribution, and use of energy. Enzymatic processes use up a lot of energy. This relates to our lab and BRCA because BRCA codes for proteins that fix damaged DNA. These proteins are enzymes and they use up a lot of energy. The BRCA mutation leads to persistent DNA damage. The image shows that the BRCA gene leads to DNA repair. As you can see in the image, the BRCA gene leads to the creation of proteins like RAD51, MRE11, NBS1, and RAD50. These proteins then repair the damaged DNA. However, if there's a mutation in the BRCA gene that makes it unfunctional, then this gene cannot be transcribed to create these proteins which fix the DNA, so the DNA doesn't get repaired. Unit 4 is all about cell cycle and cell communication. This unit focuses on regulation and communication of the cell cycle. BRCA allows damaged DNA to pass through the S checkpoint. Women with BRCA are at risk of cancer. The BRCA mutation causes genes to not fix DNA. This image shows a lack of an S-phase checkpoint, which leads to uncontrolled cell division and cancer. If you look at the image, if BRCA is not fixing DNA, then damaged DNA is going through the S phase and is being replicated, which can lead to cancer. Unit 5 is on heredity. This unit focuses on how genes are passed on throughout families. You inherit one copy of each BRCA gene from each parent. Our lab focused on heritability of the BRCA mutation. The BRCA mutation causes higher chance of developing cancer due to the fact that a mutation in BRCA leads to DNA not being repaired properly when it's broken. This image shows the pedigree of the Alterman family from the lab. As you can see, the highlighted yellow means that they have breast cancer, meaning they have the BRCA gene. The whole point of the lab was to determine if Hannah and Sarah, and maybe even Rachel's children, had the BRCA mutation. As you can see here, Deborah Alterman, Hannah, Sarah, and Rachel's mother, had cancer. Her father also had cancer, and Rachel had cancer. Hannah and Sarah haven't developed cancer yet, but there's a possibility they could in the future due to having the BRCA mutation or not.
Unit 6 discussed how genes are expressed, especially relating to regulatory mechanisms. For eukaryotes, this is done through chemical activators that are only present in certain areas of the organism so that only certain genes are expressed in those areas. The problem is mutated BRCA genes cannot be properly expressed. Therefore, the BRCA proteins will not function. And without the BRCA proteins, this allows mutations to get through and could cause a buildup of extra cells which cause tumor and could cause a cancer. The picture illustrates all the functions of the BRCA gene and all of the chemical activators required to make sure that it is transcribed for those purposes.
Unit 7 focuses on evolutionary biology, examining mechanisms such as natural selection, genetic drift, gene flow, and mutations. Hereditary breast cancer, which makes up to 5 to 10% of cases, is often linked to mutations in the BRCA gene. Inheriting a mutated BRCA gene significantly raises the risk of developing breast cancer, especially if multiple family members are affected or have been affected. Understanding gene flow helps explain how BRCA mutations spread within and between populations. Studying the BRCA gene reveals genetic differences and provides insights into the evolutionary dynamics of disease susceptibility, highlighting the principles of evolutionary biology. Unit 8 covers ecology, which focuses on gene flow, energy flow, cycles, and biodiversity. Chemicals from industrial processes affect air, water, and household environments. And some of these chemicals, like DDT, PCBs, PFAs, and fire retardants, are linked to an increased risk of breast cancer. While only 10% of breast cancer cases are due to genetic factors, BRCA mutations play a significant role in these situations, contributing to genetic diversity within populations. This can explain how diseases impact population genetics and reveals the complex relationship between environmental factors and genetic variation. Understanding these dynamics is crucial for addressing the ecological and health effects of environmental exposures and protecting biodiversity and health.
Here are the two different sides behind the bioethical concern of sharing knowledge of the BRCA mutation. The BRCA mutation causes high risk of developing cancer because the BRCA gene encodes for proteins that fix DNA. So when this gene is faulty, these proteins are not made and they cannot fix DNA. This can lead to a higher risk of developing cancer. The BRCA mutation is also genetically inherited. Perspective 1 is that you should tell your spouse because it could have an effect on their future children being more likely to develop cancer. However, perspective 2 may argue that you shouldn't have to disclose this information because it could negatively affect relationships and that it may not even affect the children in the future because it's not a sure thing that they would develop cancer or even inherit the mutation.
The bioethics of genetic testing are a subject that is continuously debated and hotly contested. Genetic testing has innumerable uses throughout much of our lives, such as preventing disease and diagnosing disease, as well as in the commercial world, identifying relatives and your own family history. Some negative possibilities of genetic testing include insurance implications, such as increased premiums for health and life insurance. Could impact your job security, as well as overall mental health if you find out that you have a possibility for a deadly disease. But it could also better your mental health by knowing that you might not have, you don't have the possibility for the disease. Its main use is prevention, identification, and peace of mind surrounding your genes.
Ashkenazi Jews have a higher BRCA mutation rate, which could cause cultural sensitivity. From one perspective, increased awareness and early interventions can help aid people, as testing can save lives by identifying those at risk and allowing for preventative measures. From another perspective, there's potential stigma, as individuals may face discrimination due to their genetic predisposition. This may impact families and marriage prospects, leading to social and cultural challenges. Addressing these concerns requires a balanced approach that promotes awareness and health benefits while protecting against discrimination and respecting cultural sensitivities.